Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 4 for January 2025. Let's begin with the first question. In question 1, the diagram shows some processes linking photosynthesis to productivity and the distribution of biomass in the trees in a forest. So this is what they've given us, the different boxes. And they say which row of the table identifies the box that represents net primary productivity, NPP, the box that represents gross primary productivity, GPP, and the box that represents respiration, R. So from photosynthesis, we will create the GPP, and then when respiration goes away or as a loss from respiration, the remaining part is going to be NPP. So if we look at the answers here, the possible answers, it's only C that matches. Box J is GPP, then we see R, and then the last one is NPP. Next, they say which are suitable units for NPP. Now, our net primary productivity is going to be measured in amount of energy per area per time. So we can see it should be year, not year squared. These two are out. And here we can see per m, it should be area squared. So area or meter squared, which is area. So that's out, leaving this kilojoules per meter squared per year as the answer. In part C, they say the biomass distributed to the leaves, wood and roots of the trees in the forest or in this forest was 12,500. The biomass was distributed 34% to the leaves, 37% to the roots, and the rest to the wood of the trees. Calculate how many units of biomass were distributed to the wood of the trees. Now, because they say the rest, we need to subtract this from the total, which is 100, in order to get the percentage that goes to the wood. So the percentage should be 100 minus the sum of these two, giving us 29. And so to calculate how many units are distributed, it's going to be 29 divided by 100 times the 12,500, and I got 3625. That is my answer. Next, they say which row of the table shows what can be found in the biomass of leaves and roots of trees. So from the possible answers, in the leaves, we expect there to be chlorophyll and of course starch. My answer was C, chlorophyll, starch in the leaves, and then starch protein in the roots, as proteins are required for growth and increase in size all over the plant. Next, they say forests are examples of ecosystems. State what is meant by the term ecosystem. This is easy. An ecosystem could be a community of organisms interacting with each other and with their physical environment. The physical environment could be the abiotic, meaning the water, the land, and so on, in a defined area. In question 2, they say the time of death of a mammal can be determined in a number of ways. The identification of insects present on the body can be used to estimate how long a mammal has been dead. Which method uses the numbers and species of insects to estimate how long a mammal has been dead? That is forensic entomology. It studies insects. Then they say explain why body temperature and the degree of muscle contraction can be used to determine the time of death of a mammal. We know after somebody dies, the body temperature is going to decrease and the muscle contraction, or the muscles, are going to be in a contracted state a few hours after death. So due to the variation in muscle contraction, usually a few hours after death, we can use this muscle contraction as a predictor. And the temperature, we can study that based on the ambient temperature. So I said body temperature begins to decrease after death in a predictable manner due to the absence of metabolic heat production. This drop follows a curve, a cooling curve, typically decreasing by about 1.5 to 2 degrees per hour under normal conditions. By comparing the measured body temperature to normal living body temperature, which is usually about 37 degrees, and accounting for environmental factors, an estimate of the time of death can be made. Muscle contraction refers to rigor mortis, which is the stiffening of the muscles that begin a few hours after death due to lack of ATP. Rigor mortis follows a predictable timeline, and it helps to estimate the time of death, especially when used alongside body temperature. In question 3, the photograph shows a Canadian lynx and a hare, so we can see the two. They say a lynx is a type of cat and is a predator of the hare. Graph 1 shows the changes in the number of lynx from 1845 to 1895. So on the vertical axis, we see the number in thousands. And then on the horizontal axis, we see the years. So they say in 1863, there were 150,000 hairs. Calculate how many more hairs were there than lynx. So I went to the graph to see at 1863, and the corresponding number was about 20. We can see it there. So since it's in 1,000, so 20,000. And this one here was 150,000. So 150,000 divided by 20,000, I got 7.5 times as my answer. Then next they say graph 2 shows part of a graph. They say sketch a line on graph 2 to show the likely changes in the numbers of hairs from 1855 to 1880. Now, because this is a predator-prey relationship, we expect, if you go back here, they told us the lynx are predators of hares. So since this is showing us the population of lynx, we expect when the predators are going down, the number of prey is going to increase. And of course, the total number of prey should always be higher than the predators. So they increase, and the increase is going to be higher than the peak of the predator. So they increase. And then as they reach their maximum, these are trying to increase up as well. And then when they increase, of course, they're eating the prey number is going to decrease and so on. So it's an oscillation curve, a typical example of predator-prey relationships. So then they say explain the changes in number of lynx and hares. So the changes we see, of course, are like I've already said, it's a typical predator-prey relationship. When the hare numbers increase, there is more food for the lynx, so the lynx numbers increase with a slight delay. As lynx numbers increase, they hunt and eat more hares, so the hare population declines. Fewer hares mean less food for the lynx, causing starvation and a drop in the population of the lynx. That's why we see when the hares are decreasing, of course, this is because they're being eaten, and when they are at their lowest, we see these begin to decrease because there is no food for them to eat, and that gives time for the hares to begin to increase as well. Then next they say in many ecosystems, the relationship between one predator species and one prey species is less predictable. Suggest 2 reasons why this relationship is not predictable. This could be because there are other factors that could affect the numbers of the prey. For example, some predators have different prey. And another thing is there are some other factors like disease that could affect the numbers. So I said other food sources may be available for the predator, reducing its dependence on one prey species. The numbers of prey depend on availability of their food, and also environmental factors like disease, weather, and habitat changes can affect the populations independently of predation. In question 4, the structure of a chloroplast is related to its role in photosynthesis. So here we see the structure of the chloroplast. We see the thylakoids, the thylakoid space, the stroma, and so many different things in here. Then they say which row of the table shows where the light-dependent reactions and the light-independent reactions take place. The light-dependent reactions have to take place in the thylakoids, and the light-independent should take place in the stroma. So we can see it's going to be T and R, which is answer C. And next, where do hydrogen ions, which are the protons, accumulate? They accumulate within the space of the thylakoids, which is going to be S. So that's my answer here. Next, they say the diagram is 16,000 times larger than the actual chloroplast. Calculate the surface area of the actual chloroplast using this formula. So from the diagram, I got a measurement of 9.6. This is in centimeters, but remember they've given us, we have to write in micrometer squared, so I need to convert this into micrometer, kind of the diameter into micrometer. To convert centimeters to micrometer, you multiply by 10,000, so I got that. And so I needed to get the actual. The actual is going to be that divided by the 16,000 because they say 16,000 times. And this gave me a 6 as my diameter. So to substitute into this formula, 4 pi r squared, 4 times pi, radius is going to be 6 divided by 2, which is the same as 4 pi 3 squared. And I got 113 micrometer squared since they wanted us to give the answer as a whole number. Then next I say explain the advantages of thylakoids in a chloroplast having a large surface area. If there is a large surface area, then there is more chlorophyll, more photosystems, and so there is going to be more NADP or reduced NADP that is going to be formed and more ATP. So I say the large surface area provides more space for photosystems and of course for chlorophyll, ATP synthase enzymes, and the electron carriers involved in light-dependent reactions. This increases the efficiency of light absorption and production of ATP as well as reduced NADP. In part B, in the light-dependent reactions, ATP and reduced NADP are generated. These products are used in the light-independent reactions. The diagram shows some of the light-independent reactions. We can see this is a portion of the Calvin cycle. Then they say which describes how ATP is generated in the light-dependent reactions. So in the light-dependent reactions, ATP is going to be generated as ADP is converted to ATP. So basically phosphorylation of ADP, and the answer here should be a D. And next they say when one molecule of glucose is made in the light-independent reactions, the rate of ATP used to reduce NADP is 1.5 to 1. Show how this ratio can be calculated using the information in the diagram. When we go back here, we can see these are 3 carbon dioxide molecules, so we are going to need 2 cycles in order to produce glucose. So if we need 2 cycles, it means the ATP used per cycle is going to be the 6 plus the 3, giving us 9. So for 2 cycles, it's going to be 18 ATP molecules. And the reduced NADPs that are used are going to be 6 per cycle, so total is going to be 12. And so my ratio is going to be 18 to 12, which is 1.5 to 1. Explain why only a small proportion of the GAL produced is used to make the glucose. This is because we have to regenerate root P, so I said more scalp is used to regenerate root P so that the Calvin cycle can continue for more carbon dioxide to be fixed. In question 5, they say peat bogs are waterlogged areas with low pH. Pollen grains in peat bogs can be used to determine the types of plants that grew in a particular area in the past. The diagram shows the types of pollen, meaning the shaded areas found in a site in New Mexico. Cereal crops and beans are a source of food for humans and their animals. So we can see this is a number of years ago, and of course 4,000 years ago we had abundance of trees, and over time these are decreasing, and that decrease appeared at the same time we see the cereal crops began, because this is due to farming, and as beans came up we see a further decrease. So this is for consumption, meaning humans are cutting down trees and they are planting these crops, so the number of trees are decreasing. They say explain why the abundance of these types of pollen changes as shown in this diagram. So I say tree pollen is more abundant in earlier layers because fewer humans lived in the area and there was more forest. As humans began farming, tree pollen decreased while cereal crops and bean pollen increased due to the deforestation and agriculture. Cereal and bean pollen became more abundant as farming became established, showing the shift from natural woodlands to farmlands. In part B, they say in 1950, the preserved body of a man was found in a peat bog in Denmark. The man died over 2,000 years ago. The graph shows the maximum and minimum mean temperature and the mean rainfall in Denmark. So we can see the different data here. And they say which is the difference between the maximum and the minimum temperatures in May. When we come to the month of May, we see the maximum is about 16, that's 16 actually, and the minimum, which is 8. So the difference should be 16 minus 8, giving us 8, and so the answer is 8 degrees. And they say explain how the conditions in the peat bog preserve the body of this man. Use the information in the question and your own knowledge to support your answer. We know that peat bogs have low oxygen, there is going to be a low pH, so the decomposition, even the decomposing microorganisms cannot function properly, so it's going to be a slow decomposition process. Also, the bog is going to protect the body from being broken down or either eaten by scavengers. So I said peat bogs are waterlogged and anaerobic, meaning there is low oxygen, which slows or prevents decomposition. Low pH, meaning in acidic conditions, inhibits microbial activity and enzyme action that would normally break down tissues. The low temperature also reduces the rate of decay by slowing down enzyme activity, so less ATP released from aerobic respiration. Lack of oxygen prevents aerobic bacteria and fungi from growing. and the bulk protects the body from other organisms like scavenging animals and insects. These factors together create an environment that preserves soft tissue, skin, and sometimes even internal organs. In question 6, the development of antibiotic resistance in bacteria is a global concern. A new type of drug is being developed against bacteria. This drug has a number of effects on bacteria, including making the bacteria more susceptible to antibiotics. This drug is not an antibiotic. State the meaning of the term antibiotic. Antibiotic is a substance that kills or inhibits the growth of bacteria without harming human cells. And next they say in 2019, 1.27 million deaths were due to antibiotic resistance. It is estimated that these will rise to 10 million per year by 2050, which is the percentage increase in the number of deaths due to antibiotic resistance. So here it's going to be 10 million minus 1.27 to give us the increase, divide by the original times 100, and it's going to be 687%. So in part C, they say explain why it is important that these new types of drugs are developed. This is because bacteria are getting resistant to some other antibiotics, so we need to create new ones or to develop new ones so that we can overcome this resistance, also that we can cure some diseases because the bacteria will not be resistant to the new ones. So I said bacteria are evolving resistance to existing antibiotics, reducing their effectiveness. Bacteria are not resistant to the new drugs. These are needed to restore antibiotic effectiveness and treat infections caused by the resistant bacteria. Then they say the diagram shows the structure of the cell wall of a bacterial cell. It also shows the cell membrane. The molecules in the outer membrane vary in different types of bacteria. So we see there is an outer membrane, the peptidoglycan, we see the cell membrane, and then the cytoplasm. They say when bacteria are treated with a new drug together with the antibiotic, the antibiotics were effective at low doses. The new drug attached to the membrane or to the outer membrane suggests why the new drug resulted in antibiotics being effective at low concentration. This is because if the drug used attaches to the outer membrane, maybe it makes it more permeable to the antibiotics so that it's going to be effective even at low concentration. So I say the new drug attaches to the outer membrane of bacteria, making the membrane more permeable. This allows more antibiotic to enter the bacterial cell, increasing the effectiveness even at low concentrations. The graph shows the effects of 3 antibiotics on the species richness of gut flora. The graph shows a decrease in the species richness with the antibiotics use. So we see antibiotic C was more effective at decreasing the species richness, meaning it killed more from the gut in comparison to the other two. So they say describe 2 conclusions that can be made about the effects of these 3 antibiotics on the species richness of gut flora. This one showed the greatest decrease in the number of species in the gut flora, and A showed the least, but we can see from here up to until 3 days of taking the antibiotics, we can see there is almost a similar effect. So I said all 3 antibiotics reduce species richness in the gut flora over time. All of them show similar effectiveness for the first 3 days, but C caused the greatest decrease in species richness, followed by B, and then finally A. Next, I say explain the advantages of adding this new drug when treating a bacterial infection with antibiotic C. So like we already saw, when you use this new drug, it's going to kind of alter the cell membrane, improving the intake or uptake of the antibiotic. This enhances the effect of antibiotic C, meaning a lower dose is going to be needed, and there could be faster recovery time. Also, side effects could be reduced. It can also protect gut flora from being affected so much, enabling them to outcompete the pathogens for space and nutrients. In question 7, carbon dioxide, methane, and nitrous oxide are greenhouse gases. Nitrous oxide is produced by bacteria in the soil. State 1 anthropogenic source of carbon dioxide and 1 different anthropogenic source of methane. So carbon dioxide, of course, from burning fossil fuels, and methane from cattle farming or the rice paddy fields from where rice is grown. And then they say describe how greenhouse gases cause global warming. This occurs when the short waves from the sun reach the surface of the earth. They'll be reflected back as infrared, which is longer, and these are going to be absorbed by the accumulated greenhouse gases in the atmosphere, maintaining the heat closer to the surface of the earth. So I said greenhouse gases accumulate in the upper atmosphere. As short wavelengths from the sun reach the earth's surface, they are reflected back as infrared radiation, and this is absorbed by greenhouse gases, increasing the atmospheric temperature. In part C, they say the graph shows the changes in mean global temperatures from 1975 to 2023. So on the vertical axis, we see the changes in mean global temperatures, and down here we see the time in years. Of course, the relationship is a fluctuating one, but overall we see there is an increase. They say predict the change in the mean global temperature in 2050 from the data in the graph. So here you have to draw a line of best fit through the data. And again, this is going to be varying a little. There is going to be a little variation from the different students. Maybe you expect something to be from about this range to about maybe 1.5. Around this range is acceptable. So all you have to do is make sure the line is almost evenly going through the data so that you can get the required answer. For me, I got 1.26, but of course I was using the iPad on the PDF version. Somebody using the paper could get a different answer. This is because the PDF version I have and the paper version are actually slightly different. I realize they're slightly different because when I do some magnification calculations, I see the measurements are different, so watch out for that. But this is also acceptable. I got 1.26. In part D, they say the greenhouse gases have different warming potentials. Over a 100-year time scale, 1 ton of methane would generate 28 times the WP of 1 ton of carbon dioxide. 1 ton of nitrous oxide would generate 265 times the WP of 1 ton of carbon dioxide. So they say calculate the ratio of the WP of nitrous oxide to the WP of methane, and your answer should be to 2 significant figures. So from the data, nitrous oxide is 265 and methane is 28. So all I needed to do is divide by that. I got 9.46, and so my ratio was 9.5 to 1, and that is the answer here, of course, to 2 significant figures. And next, they say carbon dioxide can last in the atmosphere for thousands of years. Methane lasts for 12 years and nitrous oxide for 121 years. They say discuss the implications of this data and the WPs when scientists are deciding how to tackle climate change. So when we go back here, you remember we saw nitrous oxide, the WP value is 265 times, meaning this is really, really dangerous. And then we go to methane here. So in the short term, you would deal with the nitrous oxide because it stays a little bit longer, but also its WP value is quite high. Then carbon dioxide could be the last. So I said carbon dioxide remains in the atmosphere for thousands of years, so its effects are long-term. Methane has a shorter lifespan, but a higher warming potential per ton. Nitrous oxide lasts for over a century and has even greater warming potentials. So scientists must consider both how long the gas stays in the atmosphere and how strongly it traps heat. Strategies may focus on reducing the high WP gases like nitrous oxide in the short term and long-term lived gases like carbon dioxide for long-term climate impact. In part E, they say methanotrophs are a group of bacteria that can absorb methane and use it as a source of energy and as material to convert into biomass. Most methanotrophs grow best in the methane concentrations of 5,000 to 10,000. This is greater than the methane concentration in the atmosphere. Scientists wanted to identify the methanotrophs that could grow at low methane concentrations, as this could help reduce the levels of methane in the atmosphere. The graph shows the growth curve for 2 types of methanotrophs grown in a methane concentration of 500. The increase in turbidity of the culture was used to assess the growth of the methanotrophs. So on the vertical axis, they use turbidity, and on the horizontal axis, we have time in culture, which is in days. So we see that Methanotroph B showed an increase in turbidity, and actually the gradient is quite high, or you would say the rate is higher. So this explains how this graph can be used to compare the growth rate of these two types of Methanotrophs. So I said because higher turbidity means more cells, you need to draw a line of best fit for both and then compare the gradients of the two lines. Then they suggest why low methane concentrations are not able to support the growth of some Methanotrophs, such as Methanotroph A. To answer this, we can go back to the beginning here. They said the Methanotrophs, they use this as a source of energy and again for material in biomass. So meaning if we have a lower concentration of methane, then they will not have a sufficient amount to use in metabolism. So I said low methane concentrations do not enable efficient diffusion of methane into the cell, hence there is not enough energy for metabolism. And then suggest why the scientists analyzed the cultures of Methanotroph B for the production of nitrous oxide and carbon dioxide. They wanted to make sure that if they're trying to reduce methane by working with Methanotrophs, those same organisms are not producing nitrous oxide and CO2. So I say scientists analyzed the cultures to check if Methanotroph B was releasing other greenhouse gases like those, to ensure that using Methanotroph B to reduce methane doesn't cause unintended increase in the other greenhouse gases. In question 8, since the early 2020s, the number of cholera cases and deaths due to the disease are high as the world is facing its 7th pandemic of cholera. Cholera is caused by ingestion of food or water contaminated with the bacterium Vibrio cholerae. The diagram shows the distribution of cholera cases by age group and sex at birth in Malawi in the first 9 months of 2022. So on the vertical axis we see age, and the horizontal axis we see cases. Overall we see in men there were more cases than in females, and we can see the highest age group was 21 to 30, and the lowest of course is 91 to 100. So they say describe two conclusions that can be made about the distribution of cholera cases by age group and sex. This is the same thing I've said. The highest number of cholera cases occurred in 21 to 30 age group for both males and females, and in most age groups, males had more cholera cases than females. So here they say Vibrio cholerae produced a toxin made up of several subunits. Genetically modified rice plants are grown that produce rice grains that contain one of these subunits. This subunit acts as a vaccine. The rice grains were harvested and ground into a powder that can be eaten. In a clinical trial, 3 groups of people were given one of 3 doses of this rice vaccine. Another group of people were given a placebo. The change in the levels of antibody against the subunit in their blood was measured over an 8-week period, and the graphs show the data obtained. From the graph we see there is a higher change in the rice powder vaccine in comparison to the placebo, because it's like almost the placebo did not that much change. We see error bars were included, but there is overlap. Over here we see kind of something similar, changes in levels of antibodies. We see greater in the vaccine, the placebo is lower, and finally changes in the level. This is for the 6-gram rice vaccine. If you're increasing the concentration, we see more. So our summary can be like increasing the concentration kind of led to an increase in the changes of levels of antibodies. And as we go on, we see that error bars, at least here there is no overlap, but most times there is going to be overlap. In these two, between that, and between that and that, there is no overlap. So they say describe suitable placebos for use in this clinical trial. Use the information in the question to support your answer. Here they could make powder from rice grains that do not contain the vaccine subunit, and the powder should be the same concentration. It should look and taste exactly the same like the rice powder vaccine to ensure a fair test, and the participants should not be aware of what they're taking. Then they say scientists running the trial concluded that a dose of 6 grams of rice powder vaccine was needed to stimulate the immune response. State why they came to this conclusion. If we go here, this is 6 grams, we can see there is a bigger difference between the placebo group and the vaccinated group, and we can see fewer error bars overlap. Here, that and that doesn't overlap, that and that does not, but here everything overlapped, and above here everything overlapped, so we can conclude that there is a significant difference in some of the results here. So I said it showed a clear increase in antibody levels compared to the placebo and other doses. It also has fewer overlapping bars. Then they say the scientists found that these antibodies also bind to different toxins produced by another type of bacteria. Explain why these antibodies produced as a result of rice powder vaccine could bind to different toxins. They could bind to different toxins because of the complementary shapes. So I say the different toxins may have similar shapes, or antigen structures allowing the antibodies to bind to more than one type. Antibodies are specific to certain shapes on the antigens. Then here they say some people in the clinical trial did not respond to the rice powder vaccine. The scientists determined the proportion of different types of bacteria, A, B, C, D, and others, in the gut flora of these non-responders with those of responders, and the charts show the results. So here we see for non-responders and responders, and we see A was higher in the non-responders in comparison to the responders, and we see B, C, and D were higher here in comparison to that, and of course others were higher here in comparison to that. So they say compare and contrast the results for non-responders and responders. Simple, I said both had A, B, C, D, and others, and for both A was the most abundant. However, non-responders had a higher proportion of bacteria type A. Responders had greater percentage of B, C, D. You could also say and others. Give two limitations when using this data to determine if the type of gut flora could cause a person to be a non-responder to the vaccine. As you know, correlation does not prove causation, so it means that the factors could actually influence the response, and also it could be hard to identify since a combination of bacteria types can also be responsible. So next they say the human genome contains sequences of DNA called endogenous retroviruses, ERVs. These ERVs result from infection with retroviruses. Retroviruses are a group of RNA viruses that have a replication cycle similar to that of the human immunodeficiency virus. This replication cycle results in latency. Some of the ERVs in the human genome have been present for millions of years because the ERVs are passed down through many generations. This means through gametes. Explain how retroviruses introduce ERVs into human cells and how the ancient ERVs can be transmitted to offsprings in families. So here, the gamete cell is going to be infected, RNA is going to be introduced, the RNA is going to be converted into DNA using reverse transcriptase, and then that is going to be integrated into the DNA using integrase. When fertilization occurs, we will produce a zygote that has the ERV genes incorporated, and by mitosis, the embryo is going to be formed, baby is going to be born, and they will retain those same ERVs within their DNA, and since there is no selection pressure, this is not going to be expressed. So I said retroviruses introduce ERVs by first attaching to the surface of the human cells using specific receptors. Once inside, the viral RNA is converted into DNA by the enzyme reverse transcriptase. This newly formed viral DNA is then inserted into the host cell's genome with the help of another enzyme integrase, becoming part of the cell's own DNA. If this integration occurs in the cells involved in reproduction, such as gametes or early embryonic cells, the ERV becomes part of the genetic material passed to the offsprings, and at fertilization, the ERVs are present in the zygote, and as the embryo develops, mitotic division ensures that all cells inherit the ERVs. Over generations, these ERVs are passed down through inherited DNA because they often don't express harmful gene products or because expression is prevented by mechanisms like methylation or RNA silencing. They aren't removed by natural selection and can remain in the genome for millions of years. Next, they say one group of ERVs code for viral envelope proteins. In normal cells, these proteins are not synthesized due to epigenetic regulations. Explain how epigenetic regulations prevent the synthesis of these viral envelopes. So epigenetic modification could be methylation, or you could say histone modification or DNA methylation. So I say these include DNA methylation, and they can occur at the promoter region of the ERV gene, and it's going to prevent transcription factors from binding, and as a result the gene is not transcribed or expressed, and the viral envelope protein is not going to be produced. Next, they say in cancer cells, the viral envelope proteins are synthesized and incorporated into the cancer cell membrane. Explain why these viral envelope proteins could be used in a vaccine against cancer cells. So these are going to be detected as antigens. Of course, the antigens will be present. The immune system is going to be triggered. We're going to produce memory cells, plasma cells, antibodies, and T killer cells will be activated. So I said in cancer cells, the ERV envelope proteins are expressed on the cell surface. These proteins are not found on normal body cells, so they act as foreign antigens. A vaccine could stimulate the immune system to produce memory cells and plasma cells. The plasma cells produce antibodies. So this was opsonization, I've corrected that. So antibodies carry out agglutination and opsonization of cancer cells, enabling the macrophages to swiftly destroy the cells. Also, the T killer cells release bufferins that cause the destruction of the cancer cells. So this brings us to the end of question 9, as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.